somewhat repetitive, but we could still review to help you study. So a lot of you guys had questions about Hardy-Weinberg, so I want to go through a couple things with Hardy-Weinberg. Take notes on this. I will put all this stuff online. I don't have a study guide for you because we are literally just doing the study guide that I would have given you together today to review. Okay, so Hardy-Weinberg, what does it mean if a population is in Hardy-Weinberg equilibrium? Bobby, what does that mean?
They're not evolved.
They're not evolving, okay? So we would say if a population is in Hardy-Weinberg, they're in what we call genetic equilibrium. Meaning the population is not evolving, okay? Do we see this a lot in nature? No, because populations evolve, okay? So genetic equilibrium, meaning the population's not evolving. So if you have a parent population and 50%, and so let's say they're flowers, 50% of those flowers look pink and 50% of them are white. If the population's in Hardy-Weinberg or genetic equilibrium, what would we say that their offspring, let's say 8 generations down, would be? It would still be 50-50. It means it's in genetic equilibrium. The population's not evolving. Just like when you guys worked with your plants, okay, we saw that those populations were not evolving. We did a chi-square test, and we statistically supported the fact that those populations did not evolve. There wasn't a significant difference between offspring and the parents. So the population was in genetic equilibrium. That's what Hardy-Weinberg said. Okay?
This one.
Yes?
How much would you call a big discrepancy? Like if you did do
You would have to do a chi-square test. Your value would have to be higher than what your table, your p-value is in your chi-square table. Okay? Now, there's 5 conditions that need to be met in order for Hardy-Weinberg or genetic equilibrium hold true. These usually don't happen in everyday life, in nature, so that's why we usually see that populations evolve. Okay, what are the 5 conditions?
No mutation.
I wrote that number 1 too big.
Oh my god.
No. What's one, Rose?
No mutations.
No mutations. No mutations throughout the population in order for that population to be in genetic equilibrium. Okay? Nicole, give me another one.
Large population size.
Large population size.
It's like backwards. It's like backwards.
Is another one. Jenna.
Random mating.
Random mating. So one thing is not favored by sexual selection. It's random mating to create offspring. Okay, what's a 4th one? Claudia.
No gene flow.
No gene flow. What does that mean? What is gene flow?
No immigration or emigration.
Very nice. It's very specific. I like the way you define that. No immigration, no emigration. People are not coming into the population. People or organisms aren't coming into the population. Organisms are not leaving the population. Okay, there's no gene flow. There's a 5th one.
This one is.
I know this
That's another thing with population.
No. Are we level like look at our notes?
Yeah, I don't care.
No, this is the test. We're doing it together.
Stop.
Sorry.
Oh yeah, wait. Hardy-Weinberg was in chapter 17?
No natural selection.
There you go. 16 or 17. How did you figure that?
No natural selection, which is the mechanism for evolution. So obviously the population can't be affected by natural selection, okay, or it's going to evolve. That's how evolution happens. Claudia.
Alright, this may sound stupid, but isn't random mating and natural selection pretty much the same thing?
No.
No, because natural selection is like genes or alleles becoming different because of an organism is like more fit or less fit for their environment.
Yeah.
So it's not really the same thing. This is like the overarching mechanism for evolution.
Okay.
This would just be like one it's not favored by sexual selection. It's just random. Okay? So those 5 things need to be met in order for Hardy-Weinberg to hold true. If any of these things are happening in the population, this can disrupt genetic equilibrium. Okay? If there's gene flow, organisms are coming in and out of the population, that disrupts genetic equilibrium. Okay? If there's a small population size, What if those small populations have, you have such a tiny population, it just so happens that 2 of them have a mutated allele. Now your gene pool is going to not be in genetic equilibrium after that small gene pool reproduces. So any of these things can have an effect and screw up the genetic equilibrium. Okay, and the population would not be in Hardy-Weinberg. Does that make sense? Okay, now we can mathematically figure out the frequency of alleles in a population and how many individuals in a population are going to be like heterozygous, homozygous dominant, homozygous recessive. Okay, when I'm talking about an allele, how many alleles are in this? 2 alleles. So we can figure out the frequency of the dominant allele, which would be just this. We can figure out the frequency of the recessive allele, which would be just that. Or we could figure out how many individuals are heterozygous for that population, how many individuals are homozygous dominant, how many individuals are homozygous recessive for that population, by using the Hardy-Weinberg formula. Okay? Now, what's the Hardy-Weinberg formula?
E squared
Plus
plus
2pq plus q squared equals 1.
That 1 represents 100% of the population. P squared plus 2pq plus q squared equals 1. Now, anytime you are hearing someone talk about Hardy-Weinberg, which probably isn't that often, you see a Hardy-Weinberg problem in AP biology, which would probably happen more. I don't know if this thing's recording when it, like the.
When it goes off.
Yeah, I'm not sure if it's going to work or not, but whatever. Okay. Anytime you hear individuals, people, populations, okay? P squared, individuals, people, populations, are how many people are what? Dominant. How many people are homozygous dominant?
Is this the percentage of people that are homozygous?
Yes, like 65% of the people are homozygous dominant. 65 individuals are homozygous dominant. 2pq would be the number of individuals that are heterozygous for whatever trait you're looking at. Q squared would be how many people, individuals, are homozygous recessive. Okay? This has nothing to do with alleles yet. This is people, populations, individuals. Now, if we're talking about alleles, which you can also find out from Hardy-Weinberg, how many times this allele shows up in the population? How many times that allele shows up in the population? Okay, you can use P plus Q equals 1. P is the frequency of the dominant allele. So it would be like how many times this big A shows up in the population. Q is the frequency of the recessive allele. How many times this little a shows up in the population? You have to be able to make that, you have to be able to decipher between the two.
Do you have a practice problem?
I do. Any questions so far about this, though? You sure? You can move on? Okay. So, here's our practice problem. There are 150 students in a class. 96 of the students did well in the class, and the others did not.
Oh yeah, that's going to be online, right?
Yes. Let's assume this is a genetic rather than an environmental trait, which in real life that's probably environmental. And doing well in the class is the dominant trait. Find the frequency of the dominant allele, recessive allele, and number of individuals who are heterozygous for that trait. Alright. You want to talk around the phone? Okay. Sure, no problem. Hey, Rose. You know Mrs. Manick in the office? She's one of the principal, Mr. Graves' secretaries. Barb.
Just go in the main office to the front.
Yeah.
Okay.
They need to see you down there. Okay, 150 students in a class, 96% of the students did well in the class, the others did not. Let's assume it's the genetic traits. I want to find all this stuff out, okay? The first thing you should do is work with Q and find out Q, okay? You don't want to look at this and say, oh, 96% of the students, this is they did well, that's p squared. No, that's not true, okay, because that doesn't account for the heterozygotes. So what is the first thing I should do to try to solve this problem?
Track 96 from 150.
Yeah, okay. So if 150 students in the class, 96 of them did well. That's the dominant trait. That means how many of them did not do well? 54 students did not do well in the class. Now, since this is a recessive trait and you have to have both recessive alleles to have the recessive trait, we can work with this number 54. Okay? What does this 54 represent? Matt?
Q squared.
Q squared. Who agrees? Thumbs up if you agree with that. I also agree with that. Why can't this be p squared then? Once again, it doesn't account for how many heterozygotes there are. Okay? Q squared, that's the recessive. You know that they have that allele. Wait 1 second, Alex. I got to give you a pass. Perfect. Okay, so 54 would represent Q squared. Have we found Q yet?
No.
You always have to find Q. How do I find Q?
Square root.
54.
Square root it.
Wait.
What?
Don't you do
You
54
have
divided by
54 divided by
150?
150, you get 0.36, and then you square root it.
Yeah, yeah.
You have to,
54 divided by 150.
you have to get your percentage, because Q squared is a percentage of people.
Yeah,
Yeah.
yeah.
You can't just have 54.
Because 54,
54 would be,
is something squared that equals 1 that doesn't work. You have
Right,
to
wait,
divide it by
wait,
150
wait,
to
wait.
get the...
Yeah.
150 students. Yeah, you have to get the percentage. I'm sorry.
You should be. You shouldn't be on that.
54 students out of 150. That means what percent of them are?
That's 0.36.
Okay. Now this equals what?
Q squared.
Q squared. Sorry, I forgot to do the percent part.
Okay.
Okay.
Alright.
You were testing us to make sure we knew what we were doing.
I don't know
Exactly.
about
Then you do. Okay. Now how do we find Q?
I square root it.
You got to square root it. Exactly.
that. 0.7. I won.
So Q equals, what is it?
0.6.
0.6. Is
That's,
that true?
you might don't believe me.
Yeah, it is.
I do believe you. I just want to make sure we get some cross-referencing of our data here. Okay, so Q squared equals 0.6. Now, yes, Nicole?
So the 0.36 equals Q squared and not like 54.
Yes, because we have to do percentage. I'm sorry. Because if this was out of 100, you could do that, but it's not out of 100. So, you know what I'm saying? You need a percent. The original problem was out of 100, but I thought that was too easy, obviously, because I didn't even think of that. Okay. Okay, so Q is 0.6. Now, this is what? What did I just figure out?
Q.
What does Q represent?
The other part of 1.
Recessive.
One more time, Vanessa.
Frequency of recessive.
There you go. You just found out the frequency of the recessive allele. So, done that. How do I find out the frequency of the dominant allele?
Subtract that from 1.
Exactly. If P plus Q equals 1, that means P plus 0.6 equals 1. P is going to equal,
0.4.
what is it?
4.
0.4. Does that make sense?
That's
Yes,
right.
it does. Okay. That would be the frequency of the
Dominant.
dominant allele.
Yeah, that's true. I did.
Very nice. There we go. How do we find now the number of individuals who are heterozygous for the trait?
2PQ.
No.
Exactly.
Yeah.
2PQ gives us number of individuals, population people. Okay, so 2PQ, oopsie. 2 times P, which we found out was 0.4, times Q that we found out was 0.6.
0.4.
Yeah, 0.48 times 150.
0.48. That means 48% of our population is heterozygous for the trait. Now, does that tell us the number of individuals? Not necessarily. You have to take 48% of 150 people. Okay?
72.
So 48% of 150 is
72.
what 72?
72.
72 people in that population are heterozygous for that trait. Now, if you did the same thing with P squared and Q squared, you would find that all these numbers are going to match. The final people are going to equal 150 people. The percentages are going to equal 100%, and you can actually check your work by doing that. Okay? You have to find Q first. I don't know, the problem might give you, say the frequency of the recessive allele is this. Okay, then just use that. All right, this is one where you have to kind of go around to find that information. Are you guys still confused on Hardy-Weinberg?
No, man.
You sure we don't need to do another problem?
Do you have another one?
Not right here, but I could look for one.
No.
Maybe at the end of class we have time on the hard one.
Ladies, how do we feel? You guys are being kind of quiet. Madeline, how do you feel? Feel fine.
I
Let's move on. Okay, so we're good with that.
Is that all the hard
That's all the Hardy-Weinberg I have for you.
Wasn't that?
Okay, next thing here.
Oh.
There's questions about types of selection, gene flow, genetic drift, bottleneck founder effect. Some of the stuff I wrote on here for my own good. Chapter 7, what does it say? Most of the question. No. You in trouble?
It's changed.
It's been a field trip.
Okay. Chain of being, no one cares. Actually, I mean, we care, but not right now. Don't care. Not important. Not that important. Already explained genetic equilibrium. Mutation rate. You guys should know what mutation rate is. This is all online, by the way. Probability of a gene mutating during or in between DNA replications. Okay. It's important that you know that scientists feel that they can measure mutation rate in a consistent way, meaning that mutations show up in populations consistently. Okay, so that's like kind of that's part of the evidence for evolution. Okay, that mutations are consistent. It's not a big point; it's just a small little point here.
Wait, what?
Mutation rate is consistent. We can measure the mutation rate of certain populations. Okay, that's not that important.
Wait.
Apparently it is.
Guys, this is already online.
I know, but it helps to like clear it.
You know what? Yeah, you do your thing. Whatever you want. Whatever makes you feel better.
I see this
Consistent in
You got there, fruit snack?
Probably.
Oh wow.
What's up, Claudia?
Okay, I need to
So
put
you
that
have
one
to
in.
describe like the theories of the scientists like Lamreich, Matthias.
It's not in your learning objectives. You have to look at the learning objectives big picture. But no, you don't have to do that.
Such as Darwin and his natural selection.
You should, yeah, I would use that one.
So would you say if we went through all of the learning objectives and were able to answer all of them, we'd be completely set?
Yeah.
I mean, the learning objectives are big picture things. So if you can explain every single learning objective, then yeah, you would be set because that would take you a long time to be able to explain. Like, show that there's evidence for natural or if there's evidence for evolution. That's in your learning objectives. You know what I mean? So there's a lot of different things that go into that. But yeah, that gives you the best big picture ideas of what you need to know.
Wait, so if you explain learning objective with like examples and like vocab and everything, like it would be fun. Okay. 'Cause I'm about to go home and
Theoretically
memorize
speaking, yes.
it. I'll go home and memorize
She will though.
it.
She
I'm
really
going to
will.
go memorize
It's not about memorizing. It's about actually learning.
Like, I'm so scared about studying for this test. No idea. Do we have to know about polymorphism?
No.
Okay.
Gene flow. This is important here. Guys had questions on gene flow. This is the physical flow of alleles between populations. This is important. Gene flow is only because of immigration or emigration. Gene flow and genetic drift are not the same things. Okay, and I'll explain the difference between the two. Okay. This is only, gene flow is only because of immigration or emigration, the physical flow of alleles. Okay, genes are being swapped because of that. Separated populations become genetically similar because of gene flow. Okay, if you have two separate populations of organisms, but they're leaving and coming into one another's populations, and reproducing with one another, obviously their populations are going to become genetically similar to one another. Okay, if they're coming in and coming out and reproducing. It's important that you know that gene flow counters genetic drift. And I'll explain that in a minute. Gene flow counters genetic drift. Genetic drift, those populations will become separated.
Right.
Okay? But if you have gene flow, it counters that separation because they're moving.
Oh,
They're coming in and out. You know what I mean?
Yeah.
They're not the same thing. They're related, but one of them counters the other one.
On the website, we're gonna see
Would you
your.
like me to go refill your water?
I would really like that. Yeah, on the website, what?
Are we gonna see like your underlines and everything? Like you're not gonna use them.
No, I already put this on the website. This is already on there. I think I think I said on there from the beginning.
Can you explain the difference between gene flow and genetic drift again? Sorry.
No, that's fine. Genetic drift would be like two populations becoming separated from one another,
Okay.
like founder effect or something.
Yeah.
Gene flow counters genetic drift because even though those two populations are separated, if you have individuals emigrating or immigrating out of those two separated, barriers or whatever, they're going to reproduce with each other and their genes are going to mix. Okay, so gene flow counters genetic drift. Genetic drift, you would think, okay, these two populations, since they're so, distant from one another, they're going to have, they're going to become separate species, or they're going to have very distinct genes or whatever. But if they're coming in and out of each other's populations because of gene flow, then that's going to counter all those mutations and that, like, genetic separation. Does that make sense? Okay. So any questions about that? No? Move on? Okay. All right, this is genetic drift. This is random change in allele frequencies and generations by chance alone. Okay? There's a volcano. And now you have two separated areas. Okay? There's a hurricane. There's some freak event that's going to change the allele frequencies. Half the population gets wiped out and the other half's left. Okay, these are chance events that are going to change allele frequencies.
So, like, other than natural disasters, what could be an example of chance event?
Would you prefer us to do like natural disasters or like explain?
I mean, that's the most common example. You don't really need another example besides that.
Okay.
Or if, for some reason, a small population from a larger population got separated some way. Okay. Why would the impact be small on a larger population? Jake.
There's more organisms in the population, so the ones that die are not as detrimental.
Exactly. If there's more organisms in the population, okay, then you're not going to be left with like 3 random genotypes to reproduce, and they could all be like crazy mutated ones. Okay, the more it's like, the less is affected, the more there are. The smaller the population, the bigger the impact is going to be. Okay, if you have, let's say, fruit flies, a population of 5 fruit flies, okay? And they all show these dominant traits except for these 2. They show the recessive trait. Maybe they have white eyes or something. And then a chance event comes by and wipes these 3 out, and only these 2 are left to reproduce. Well, that changes the whole allele frequency of the population because it's such a small population that you just wiped out a whole trait. Obviously, this is on a way, way smaller scale than we would, that would actually happen, but
So, like,
it's more of a detrimental if the population's smaller.
what is considered a small population?
I can't really give you an exact answer for that. I mean, it just depends on how many organisms are available in the entire population. Are there different species of that organism? So you should just know that the impact is smaller on large populations. Okay.
Do we need to know about fixation?
Yep, that's the next one. Okay. Fixation's not that important, but okay. But that's this example I gave with the fruit fly would lead to fixation, meaning these 3 are wiped. Now you're only left with those alleles. Okay, so that's what fixation is. Only one kind of allele remain at a particular gene location, and they all become homozygous for it. Just like the fruit fly example I just gave you. Okay. So now you have fixation. Now you have a loss of genetic diversity. Okay. And your allele frequency is not going to change now unless there is some sort of mutation in the population. Or if there's gene flow by immigration or immigration. Remember, gene flow and genetic drift, they're definitely different things. My knee hurts so bad.
What'd you do?
I've been running and it seriously just kills. Like, I have runner's knee now.
Stretch.
I stretch, it's
I just
just,
stretch your knee.
my quads aren't strong enough, they said. I'm like, okay. So I'm like doing squats. I don't even know what the heck I'm doing.
Like when I need start training for running, I have like one of those knee braces.
That's what I was gonna.
And
Knee
Does it help?
No.
it does, for me, it
No,
does.
the knee braces do. For me,
Do you have one of those bands, or is it like a full,
It's
like that one of those skinny bands, or is it like a
yeah,
full
and
knee
it has
one?
like a hole here.
No, I
And it's
have.
like really tight. And
Yeah, I need to look
I
into
have
that.
knee problems. I literally have like the full like one with like the metal on the sides. And,
Do you tear your ACL?
hinges and- no, and like the hinges and stuff, and it didn't help.
I gotta look into
Oh,
some
mine
sort
is
of a
just like
brace.
a wrap.
Miss
It's
Lynn.
not
Yeah.
something
I
that you
have
smell.
like special levers. I have something wrong with my legs, like that's been like permanent, whatever. I know when I run, when I use like my running braces, it does help a lot. But to get some like,
Where do you go to get a knee brace?
I actually have
Anywhere.
a specific knee doctor.
Yeah, I mean, just going.
Literally, you
I
can
mean,
go
like if
anywhere
you
to,
want one like
sports
prescribed to
section.
you,
Oh,
you
okay.
have to go to like
Yeah,
somebody because like mine is custom made for me
Oh.
because that's like how like
yeah.
crappy my knees are. But
I just have weak leg muscles, maybe. It happens when you get old. You said doing
That's why I
things.
don't run. I use the elliptical because that helps.
That's a lot better on your knees.
Yeah. And it helps your thigh muscles.
Hope you're getting this, Rose. You need to know this for the test, Rose. Okay. Okay, here we go. Okay, bottleneck. There's questions on bottlenecks. This is genetic drift happens after a bottleneck. It's a severe reduction in population brought about by some intense pressure or calamity. Once again, probably some sort of natural disaster or intense environmental pressure. Now you're going to have a small population left. That's genetic drift. Okay. So maybe they're starting to hunt a certain species. There was a volcano. Okay, a small population is left and the allele frequency is altered at random. Now, this wouldn't be that big of a deal if there was gene flow in between these two populations. But sometimes that doesn't happen. So you do have allele frequency being altered. Okay, because there is no gene flow between the two populations that became separated.
So is it the same case with like genetic drift and the fact that? Oh wait. Well, does the severity of a bottleneck also depend on the size of the population?
Yeah,
Or
definitely.
is it just like a blanket thing?
No, that would depend on population size too.
I can't.
Don't overthink it.
I can't wrap my head.
Oh
around
no, there's a volcano. Half the population died. Boom, there you go.
That's it.
Yeah.
And then genetic drift happens because of that.
That is genetic drift. Yep. Jessica,
Why would genetic drift happen? That's the part I don't get.
why would it happen?
Yeah, like after.
Because of one of these,
So just like half of all the people are gonna move somewhere else just because.
or maybe they were all living in, think about like Pangea
But
or
isn't
something like that.
Isn't that founder?
Do you know what I mean? Like there's two,
Wait, isn't that founder?
founder is a type of bottleneck.
Okay.
The land became separated.
Yeah.
Or there was a volcano and all that rock cooled and now those two populations are separated because of that.
Yeah.
Do you know what I mean? So now you can only reproduce with those alleles over here and you can only reproduce with those alleles over here. The populations might become different.
Okay.
It might not happen, but this is how we get. Yeah. Okay, founder effect is a form of bottlenecking. This is when few individuals leave a population and establish a new one elsewhere. This is what the finches on the Galapagos Islands. This is what Darwin saw. They had the mainland. A few of those individuals left the mainland. They established their own populations elsewhere. Okay, there's different habitats. There's different things to eat on these mainlands. So the ones that were more fit for those specific environments were the ones that survived, and that's how you get all these different types of finches on all these different types of islands, okay, because of the environment that's there. All right, that's a type of founder effect. That's bottlenecking. When a few individuals leave, they establish somewhere else. What are those zones called that you can establish these new populations? Adaptive zones. See how we're relating all these things together? Now we're relating speciation and founder effect, which is a form of bottlenecking.
Adaptive zone. Wait.
I'm going to go over that too, but I'm just saying, like all these.
later on in the-
Claudia.
So would the finches that are just slightly different from the main ones be like a subspecies of that finch?
It depends. It depends if they can reproduce with one another or not. Subspecies is kind of still a debated thing. We don't know. Some scientists argue this is a subspecies. Some say, like, no, that's a separate species. So,
Okay.
yeah. Nicole.
Since bottlenecking is a form of, or founder's effect is a form of a bottleneck, is the founder's effect caused by a calamity of some sort?
Not necessarily.
There's up and
It could
down
be.
wave.
Okay.
There could be some pressure for that bird to go somewhere else, but they could just establish with their
Like
own.
environmental pressures like food or.
Yeah, that's a good one. Okay, maybe the food that's available, or they're looking for a different habitat or something like that.
Okay.
But other than that, they just decide to just up and leave for no reason.
Well, there's going to be probably some reason, some environmental reason why they do that.
Like predators.
Yeah, that could be it. Claudia?
Would, for example, people bringing like a specific type of plant or an organism to different place be a founder effect?
It's not really natural.
Or does that
Yeah, I don't know if it has to be natural to do that, because there's a couple examples that, you we brought something over to the United States to try to
Yeah.
establish a population that like got rapidly out of control.
Invasive species.
Yeah.
Yeah,
that's what
yeah.
Is
I think you can still consider that bottlenecking. Well, or founder effect, I mean.
Okay. I don't understand the difference between bottlenecking and founder's effect.
There
Is there
really
a
is no difference. Bottlenecking, founder effect is bottlenecking.
Okay.
Okay, because you're still getting, whoops, wrong way. You're still getting the allele frequency altered. Okay, but the only difference is a couple of them leave and go to somewhere else.
Okay.
This could be like no one leaves. It happens. So there's some sort of other environmental thing, like a natural disaster that makes the population kind of split off into two. So, does that make sense? Okay. All right, types of selection. I'm telling you right now, this is a minute detail, the different types of selection. You should just know them. Be able to look at a graph and say it's like this. I wouldn't have you write a 4-page essay about the types of selection, but you should know what each type is. Okay? Directional selection. Okay? Extreme forms of the allele are favored.
Oh, that's all.
Okay?
That's all I need.
Peppered moth is a good example of this. Okay, the types of moths that survived, it shifted in a direction that was consistent to the environment. Remember, the trees got all covered in soot and ash during the, come on, history person,
Industrial Revolution.
Industrial Revolution. Okay, and now the moths that were darker colored, which was a mutation, that mutation became beneficial.
Does it have to be a mutation?
Usually, that's what it comes from. Okay, it's not like, oh, I'm a white moth, but now I'm going to change to be dark colored so I can survive. That cannot happen. You cannot just change what you look like to better fit your environment. The ones that looked like that on accident are the ones that survive. Those survivals reproduce with one another. That's how you get all the dark colored moths. Okay, so the extreme form of the dark moth was favored. Not both extremes, just one extreme. Okay, and it was the darker phenotype that was favored here. Okay, and it was consistent with the environment. Those darker colored moths have more fitness, so they can reproduce because they're better fit for their environment. Questions on that one? My stomach is seriously growling. Next one, stabilizing selection. This is when intermediate forms are eliminated from the population. Okay, so you have extreme forms being eliminated. The most common phenotypes are favored for. Okay, there's an example from your book, I think if I'm remembering this correctly, where the fly was like medium size was the one that got favored. It was too small, it was eaten by an insect. It was too big, it was eaten by like a lizard or something. So the one that was in the middle was too small for the one, too big for the other one. So those are the ones that survive. Now those are the ones that reproduce. Rose.
See, like, I understand directional and stabilizing, but, like, with the most common phenotype, like, if the extreme, like,
Like if this is a black colored, no, let's not do color. If this is a large beet, what the heck am I doing? If this is a large fly, and this is a small fly,
and then it's like
the one intermediate right in the middle, medium fly, it's like a hybrid of the two extremes, is the one that is favored for.
But, like,
Yay.
the most common phenotype is the small one.
Because if it's
See, like, I understand the actual definition when you say most common phenotype.
Why does that mess you up?
Because, like, what if the most common phenotype is a small one?
like it's not written.
Well, these, it usually, the most common is in the middle, because these are usually like the extremes,
Okay.
where
So,
you have
So,
like,
like,
Is it safe to say
with height,
Like- dominant, recessive, heterozygous?
so, like, if
Is
you use
that-
the heterozygous.
not necessarily, because sometimes heterozygous and dominant homozygous show the same phenotype. But this you can't go straight dominant recessive with this. It's like your height. There's a lot of factors that are involved. Okay, most people in the world aren't, they don't look like freaking
So
Yao
it's-
Ming,
so
and
it's
most
kind
people don't look like
Okay, is this
are as small as
is this
some of you in here.
is this a correct
Okay.
analogy? Like
Yeah, not everyone's Alexis or Yao Ming. Most people are like Paige.
Is this a correct analogy when like buying
Very consistent height. I'm too short.
like buying clothes, like they don't like there's like the really small size and the really large.
But most people are just in the middle.
And that's why they make
Exactly.
most of those. Okay.
Exactly.
Oh, think of it that. Think of it in clothes.
Yeah.
There we go.
Whatever. Whatever helps.
Stabilizing.
That's stabilizing.
Stabilizing
Next
stitching
one.
clothes.
Disruptive
Done.
selection. Okay, both ends of a range of variation are favored. The intermediate forms are selected against. Okay, so once again, if you have this bell curve, okay, intermediate is selected against. So extreme ones are favored. So really, that wouldn't be a good one for it. It'd look like this. Okay, selected against, extreme, you're favored.
What's an example?
There is none.
Like you could use the fly example, but now let's say the predators can only get, like, the small ones are too small, the big ones are too big, they only like to eat the ones that are medium sized. Do you know what I mean? So the medium sized ones get,
Can we
it just
use that on our
depends.
test, or we're gonna use
Yeah, but I don't, yeah, you can.
Rose.
What's the difference between disruptive and directional?
Both extremes are favored for this. For disruptive, directional, only one extreme is favored. Only the dark moth was favored. Okay, for the directional. Claudia?
For our test, if we'd have to explain a type of selection, could we give like a hypothetical example, or do we still have
You can
to
give a
use some
hypothetical.
that's-
Yeah.
Can I describe it in clothes?
No, make it scientific. So I was at the mall. Okay, sexual selection. You guys know sexual selection is because females usually prefer whatever crazy thing that the bird's doing with its wings, whatever. Okay, the females are usually the ones that select it, but there's different mating rituals and stuff like that. So that would be sexual selection, non-random mating.
Advantageous.
I can't believe I'm going to talk for this whole period.
Did you not want to talk to someone?
No, I mean I didn't think that I would.
Okay, we have half an hour left, and we're not even through chapter 17.
We're done with chapter 17 now.
One and done.
Most of your questions, well, that's not true. You didn't have a lot of speciation questions.
Yeah.
What about the temporal and what was that?
We're gonna
That's chapter 18.
yeah, we're gonna go over that.
What was
Wait,
that
this
called?
is just 17?
Like isolation,
17.
temporal isolation.
Those are just types of isolation.
Yeah, those are like pre-zygotic isolating mechanisms.
Shoot, the test is only 3 questions. These are gonna be like...
Is
Okay.
How many questions is the test? 3?
It's 3, but it's like number 1 parts A, B, and C. Number 2 A, B, and C. 3 A, B, and
I
C.
love that.
Yeah,
So
there's
can
some
you-
graphs on it too. It's gonna be fun. Okay,
Can you
next
please-
one here. I already explained
I
that.
don't know if you just did it, but can you...
Okay,
line.
What?
Can you post like your writings online if you didn't just erase them?
She didn't. She said she's
Let's
gonna
post
post
some.
all that.
All right. Okay, I will. All right.
It only is
Speciation
on
review
one.
under
No, but I want
speciation
the one
folder.
for
Good
You
lord.
haven't recorded.
Jack.
Stop talking to me. Stop. I know, me too. Trust me, me too.
I know, right?
Okay. Remember, these are pre-zygotic mechanisms for speciation. What does that mean?
Before
The
the
zygotes form.
Yeah, but what does that mean? A pre-zygotic species, like isolating mechanism.
So they can't even make.
Be born.
so it's there are mechanisms that prevent the zygote from happening based on like environmental things of the parents.
Okay, so what does this cause?
Speciation.
Which is?
Genetic isolation. Or reproductive isolation.
Yes, two species. They're being reproductively isolated from one another. There can be all these different types of mechanisms. Okay, give me an example of one of these and tell me which it is.
Behavioral, when they have like different like mating rituals or something like that.
Okay, what about temporal?
Mating times of the year.
Gestation periods or different times of the year.
Yeah, you reproduce at this time, I reproduce at that time. They don't ever reproduce with one another. Two different species. Mechanical.
They can't get their sex parts next to each other.
Yes. Ecological.
I have no idea.
Someone
Forget.
knows.
Is that with the
Don't
environment?
they live in like different environments. And,
Yeah, like two species living on a mountain, but one side of the mountain's like cold and dark and under a bunch of roughage, and the other one's exposed to sunlight, and they only reproduce with those, okay? Gamete mortality, like we said, that could be a post-zygotic mechanism, okay?
Wait,
it
is or can be?
It can be. It's both.
Is there an argument?
Well,
No, most-
If
isn't it
it's after,
Yeah, isn't mostly post-zygotic? Because if
Yeah,
it's
a
formed,
gamete
then
is
I could just
post-zygotic.
yes, post-zygotic. There you go. We're not going to get into. You don't need to know.
The nitty
Okay. Allopatric speciation. Remember, there's a physical barrier between populations. It can be fast, it can be slow.
This is
Okay.
really fast.
Physical barrier in between the populations. Two different species because of that physical barrier. Okay?
When do you go back real quick?
Yeah.
Guys, this is online. There is no reason to write that down. This is already online for you. Okay? You should know the difference between allopatric and sympatric speciation. Sympatric speciation. Okay? Same land, two different species. A lot of you guys had questions on polyploidy, which is very minute detail, but still, okay? That's when you have 3 or more chromosomes that you need. Usually plants have this. That's how you get all these different species of wheat. Okay? You have more chromosomes than you need. What if those chromosomes combine, okay, together to make a new plant? There you go. There's a different species of wheat. Okay? So it's the same land, still 2 different species. Okay? I know I was talking about that. Those 2 different populations of fish, or there's a bunch of different populations of fish in 2 different lakes, 2 different species. Okay, why are they in 2 different species? Maybe because it's by chance, sexual selection. They all have a common ancestor. Okay, but basically, same land, 2 different species. Parapatric. Okay, a lot of you had questions on that. Remember, this is when you have overlapping zones of 2 different species. One species of bird here, one species of bird here. Geographically speaking, there's an overlapping zone here where the species are. In that zone, you're going to find hybrids, okay, like a mixture of the two species here. Okay, that would be parapatric. Now, once again, there might be an argument: is this a subspecies? Can they reproduce with them? It can be argued, okay, if you're a biologist. Yes, you are. Okay, peripatric. This is a type of allopatric speciation. Okay, this just happens when one or two when the populations diverge. One of them is very small. It's the same thing as allopatric speciation, but the only difference is one of the populations that diverges is smaller than the first one.
Does parapatric speciation and founder effect go hand in hand?
Yes, definitely. Matt?
Would that be the finches?
I don't know how small those populations were, but yeah, that definitely could have, one of the populations could have been very small. So it's allopatric. It's also parapatric. Parapatric is just more specific of a type of allopatric speciation when one of the populations becomes small.
Why couldn't it be sympatric as well?
Because if it's the same land, it's not sympatric speciation. If they're in the same land, they're not geographically isolated. It can't be sympatric speciation.
But
it has to be geographically isolated for these.
For parapatric?
Yes, because it is allopatric.
Wait, is Sympatric same land or different land?
Sympatric is the
Same land.
I don't get why they diverge if they're on the same land.
Yeah, that was.
That's what I was talking about. It could be due to like sexual selection. There could be,
Okay.
you know what I mean? There could be a mutation, and they diverge like that. Okay, cladogenesis and anagenesis. This is just if you look at like an evolutionary tree. Okay, this means that they gradually evolve, like branch like that. Anagenesis is when it goes like that. Okay, each of these go with. I'm going to explain what each of these go with in the next thing, but this is when they accumulate subtly. Cladogenesis is when they gradually evolve. Okay, once again, this is the graduated model of equilibrium. If you hear someone talk about this, this would be a punctuated model of equilibrium. Okay, it appears suddenly. 46 minutes.
Are you gonna be able to send that to me?
How? I would just upload that. Oh, you can't upload it to you. I don't care what you do. Okay, adaptive radiation. Many branches coming from a species. Okay, so they're bursts of divergence from one single species. Okay, they go into adaptive zones, which are habitats that are suitable for those different species. Or maybe because of those habitats that are suitable for all these different species, that's when these different species come about. Okay, but it comes from one single one.
Radiation leads to adaptive zones.
I have more about those two. Okay. And then, yeah, you don't need to know that. Okay. Okay, so we know that speciation stops gene flow. Okay, you can't have emigration or immigration. All right? So that would make them reproductively isolated, which is what you need for speciation. Okay, I already talked about polyploidy. Adaptive radiation is a penetration species. Already talked about that.
Is this the new one that you made?
Yes. Okay. So adaptive radiation is that pattern I just showed you. You get a new species from one single one. Okay, and they fill the adaptive zones, which are habitats that are evolutionary equipped to have that species thrive. Okay, speciation rates can vary. If there's a lot of habitats and a lot of adaptive zones becoming available, then you could probably have more species. Okay, if not, then you won't have as many. Okay, I'm going to post this online, so I'm going to keep going through these. Okay, moving on. Extinction. Someone had a question about extinctions. Really, you don't need to know too much about them. The extinction rates are rapid at times of ecological stress. Know that there's 5 major ones. Okay, I posted those. You don't need to know the names of them. Okay, just know that there's 5 major ones. For example, when that meteor hit the Earth and then all the dinosaurs became extinct. Okay, human impact, that can, there's endangered and extinct species because of humans hunting them or whatever. So that can happen as well. The question about radiometric dating. Now we're finally getting into macroevolution. Okay, radiometric dating is a method used to determine how old rocks and minerals are. That's how we can see how old certain fossils are. You measure the half-life. That's what measuring half-life is. It's a type of radiometric dating. You measure how long it takes for half of that sample to decay. You can measure certain isotopes in minerals, rocks. Okay, look down the fossil record, see how much is left. Then you can determine how old something is. Okay?
Are we going to have half-life problems?
No. You just need to know what that is. Okay? Couple things. Evidence of evolution. Obviously, fossils are evidence of evolution. We can see how species evolved looking at fossils. The Earth has also evolved. Just not our living species, but the Earth itself. Okay, think about Pangea. and when all the continents were together, and it took millions of years for them to split apart. That is the Earth evolving. Same thing with plate tectonics, the plates moving back and forth to make that happen. Okay? That's like some 9th grade science right there.
Yeah. 9th grade, 8th grade. For us,
Yeah, we don't
it's
teach it in
good.
9th grade anymore. I did my first 2 years. No, they teach that in middle school now. Okay, moving on. Now, a lot of questions on comparative morphology and morphological divergence and convergence. Okay, pay close attention here. Comparative morphology, think about the words. You're just comparing morphology. You're just comparing the bodies and the body structures of organisms, okay? This gives us evidence that there is descent with modification. Meaning, if I look at a bunch of structures, turn right to the page. Beast! Look at all these structures right here. Okay? Looking at these as a scientist and comparing the body structures and the body forms, that's an example of comparative morphology. Alright? How does this provide descent with modification? Well, look at these. All these different bones have the same parts, but they're modified. So they all have descent, a common ancestor, but they're becoming modified as far as like the selective pressures go. You can't just have the best arm in the whole entire universe constructed because it's the best one possible. That's not how evolution works. It has to evolve from something. And it's going to evolve gradually and be modified gradually. Okay? So that's an example of that. Now, morphological divergence. Okay, this is a pattern in which genetically diverging lineages undergo a change in body form of a common ancestor. Basically, why they're... If you look at I just showed you 1910. Okay. Why two different lineages, why their structures look the same? It's because they have a common ancestor. What type of structures does morphological divergence give you? Homologous structures, because it comes from a common ancestor. There you go. You get homologous structures from morphological divergence. It all comes from a common ancestor, descent with modification. Just gets modified. Okay, morphological convergence, on the other hand, they evolve in similar ways due to environmental pressures. So what kind of structures do you get from morphological convergence? Analogous structures. Why are they the same? Well, it's not because they have a common ancestor. It's because that they probably are susceptible to the same types of environments or the same types of pressures of, the food that they need or whatever. So structures look similar because of a common ancestor, homologous structures. Structures look similar because of environmental needs, analogous structures. Okay? Does that make sense? Yes or no? I can move on from there?
Imagine how hard AP Bio would be if we didn't have like the list of what we needed to learn.
Oh, I know. I would just make it up.
What's those things called?
Learning objectives.
Yeah.
All right, I gotta move on. I'll put this online. Okay, more evidence of evolution besides fossils. Okay, a lot of people wanted to know, but just more evidence. Early embryos of vertebrates strongly resemble one another because they inherited the same ancient plan for development. If you look at an embryo of I think this is a fish, a reptile, a bird, and a mammal, they all look pretty much the same. Okay, that shows that we came from a common ancestor, that we have the same type of plan for development. Now, why do a bird, a fish, a reptile, and a mammal all look different when they're adults? This is the reason why. It's because of transposons. That's where most mutations come from. This is parts of your genome that can kind of hop around, okay? And basically, what regulates your genes, okay, your switches and stuff like that, make the differences in adult life forms of related lineages. That's a small detail, okay? But I just think it's important to explain why they look similar when they're embryos and when we're all adults. Well, that's parts of your DNA that can kind of like jump around and be expressed differently. Okay, so throughout time, those have had an effect on how we look as adults and how different organisms look as adults. That's why, an adult pig looks different from an adult horse, but probably their embryos look the same. Okay? All right, moving on. This is very, very important. A lot of you guys are kind of a little fuzzy on molecular biology and the evidence of evolution that comes from molecular biology. Okay. First of all, DNA sequencing. Okay, what we were going to do today, which we're not going to. There's actually computer programs where you can sequence DNA of organisms and compare them. Okay, I was going to have you download those programs, and we just didn't get time to it. But that's okay. But we can expect that. What'd you say?
Are we able to do that at home or not?
Oh yeah, I'll give you what to do at home. We can expect the DNA, RNA, and proteins of closely related species to show more similarities than those of the distantly related species. There's going to be more similarities between DNA, RNA, and proteins the more related that they are. Remember, we can sequence DNA. We have that capability. We can see where, which bases line up with which bases and how much DNA is similar. Rose?
So is DNA sequencing and aligning DNA like the same thing?
We can sequence the DNA, then we align it with different organisms to see how much is similar, how much is different. The more similar it is, the more related the organism is. The more different it is, the more different the organism is.
Would those be two different evidence?
One is, you have to sequence it before you can align it. Okay. So, examples, important here. If you look at the amino acid sequence of hemoglobin, the hemoglobin protein, okay, if you look at that sequence in monkeys, humans, and chimps, it looks pretty similar. Okay, we all three of these organisms have hemoglobin. Okay, have that protein. Okay, if we sequence out that DNA and align it, we can see which one is more similar. Are we more similar to this type of monkey, or are we more similar to this type of chimp? Whatever, just by looking at that. Okay, but it's a good thing to look at because all three of us have that same amino acid sequence of hemoglobin. If you're looking at an amino acid sequence of hemoglobin and then the sequence of some other protein that not all three organisms share, that doesn't make sense to do that. Okay, but this would be a good example of something that you can compare. Okay, this one is an even better example, cytochrome C. This is a protein involved in electron transport chain. Okay, which that goes way back to earlier in the year. But this is present in a lot of species. This is present in plants, animals, fungus, bacteria. This protein is in all of those species. Okay, when we sequence it, it looks very, very similar. Okay, the cytochrome C in us, the cytochrome C in fungus look very, very similar. But they're obviously different. So you can line up and see, are we more closely related to plants? Are we more closely related to this animal? Is this fungus more closely related to this bacteria? This is why all that kingdom and those classification systems kind of got screwed up once we could sequence DNA. Because we realized that we thought these two things were alike because morphology-wise they look alike, but really they're not alike at all. Okay, because of what the DNA is. So that could be an example of something that you can actually sequence and align. Brittany, both of these things are in your book too.
So my evolutionary gem that talked about like the different proteins and whether they get like overwhelmed and stuff, they express genes differently. Does that go more along with this or with speciation?
That goes more along with evolution.
Okay.
Yeah. By the way, those evolutionary gems, you guys don't need to. I'm going to put all those presentations up. Don't spend too much time looking at those, but they are good pieces of evidence for evolution. Okay. Questions on that? Okay. You guys had questions on cladograms, which I definitely wanted to go over here.
Just a website we can write down.
All my favorites are in Chrome. Yeah. Shut up. Okay, we know that different species share. I'm going to kind of go through this tutorial with you very quickly. We know that they share different ancestors. Okay, we can construct these cladograms here. Why is this thing being such a pain in my butt? Okay. So this would be the common ancestor here. Now, where you see these little lines, these are actual traits that appear in the species or don't appear in the species. So you can look at this and say, I can say, when did, at which organism did 4 limbs appear? Or after which organism did it look like this trait left? Okay, or whatever. These all build on one another, so that's fine. Okay, but yeah, we don't need to spend too much time on that. You guys have seen this before. You know what phylogeny is. Okay, all those kingdoms, that's not in those clades. You know, everything's organized into archaea, bacteria, and eukaryotes. That's not that important. I want more. Here we go. Okay, so to understand a cladogram, you kind of have to understand like what all this stuff means. Okay, it's a lot like reading a family tree. Blah 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 blah. Remember, this is the most common ancestor. Okay, and these are all the descendants. Now, the longer the line is, the further away it was from the ancestor. So you can see past, recent. Well, this is obviously the one that's that has the amount of time that has gone by has been the greatest. Okay, so when a lineage splits like this, that's what speciation is. Okay, so we have that. Okay, unique history B, C.
Wait, where it says common ancestor of B and C?
Yes,
It's
okay,
going to
because
the same spot before where it said it was the speciation event.
same thing. I mean, that it is a speciation event, but they still come from a common ancestor. So the one that's furthest on the bottom here, that would be the common ancestor of all of these. Okay, but if one shows up here, that's not the common ancestor of A. Okay, that's a whole different species, and then B and C came from that common ancestor.
Okay, so like if you have like the slanted ones, so like the one at the bottom is the common ancestor of all of them.
Yes, if it branches to all of them, yes it is.
And if you want to find like the common ancestor between two of them, you look for the one that's the latest, or right after that.
If you want to know what the common ancestor of B and C is, you would look at that.
No, like in the line ones, the slanted.
Say it. I didn't hear your question. Say it one more time.
Like if this was E and D, and like I wanted to find the common ancestor of both, would it be D?
Like do
If
the,
you want it,
do the nodes represent a common ancestor?
this would be like, this would all come from. If that was A, that would be the common ancestor of all of those.
So like for just two of them, like the recent common ancestor.
Yes. Then like.
Like say this is C, it would be C.
Or would it be like
Yes.
Most recent common ancestor of C, if that was C, would be this one, B.
Well, like for
the most
DNA.
recent common ancestor of DNA?
Most recent common ancestor of D would be C. Most recent common ancestor of E would be D. Okay.
We can't you do both? Because we have questions about both.
Well, I mean, if you're asked D and E, then it would be C.
I believe
Yeah.
so.
I would make that
You just
like.
take the first letter and go one back. That's your most recent common ancestor.
Okay, so if you ever hear the terminology a clade, which you might hear, it's a grouping that includes a common ancestor and all the descendants in it. Okay, this is not a clade because it doesn't include all the descendants in there. This is a clade because it, yes, includes the common ancestor and all the descendants of that specific common ancestor. Same thing with there. There only is one descendant from that common ancestor. Not a clade because it doesn't include all of them here. Okay? That's really it. I'll post this website too. Okay?
Are we done?
You don't need to look at those times. Wait, I think there was something. Notice that these two cladograms represent the same thing. They're just putting it in a different way. There's not one correct way to make one. Okay? Misconceptions. Humans did not evolve from chimpanzees like a lot of people think. Okay, we all had a common ancestor, but one didn't evolve from the next. We all evolved from the common ancestor. So if you see this, humans, chimps, it does not mean we evolved, one evolved from the other one. We all evolved from this one here. Okay? Check that out.
Do you remember the cladogram website that had like a bunch of questions on it? We did it not that long ago.
I think I posted that.
Okay.
It is on.
I think that's all I wanted to show you.
When we
That was
just
important.
did with the different problems.
Oh.
I think we did. It's
Congratulations.